I thank the UMDF Power Surge organizers for inviting me to give the 2020 update on NAMDC, the North American Mitochondrial Disease Consortium. NAMDC is part of the Rare Disease Clinical Research Network, or RDCRN, and is sponsored by the NIH, specifically by the NINDS, NICHD, and Office of Dietary Supplements. Here are my financial disclosures. I'm the lead investigator of the upcoming Entrada Natural History Study of Mingi. I've received research funding from Entrada. I'm a consultant for Entrada Therapeutics and Modus Therapeutics. I'm an inventor of the patented deoxynucleoside therapy for mitochondrial DNA depletion syndrome. That patent has been licensed by Columbia University to Modus Therapeutics, and Columbia University has equity in Modus. I've also served as a site investigator for Stealth Biotherapeutics studies SPIN 300 and 301. NAMDC is comprised of 17 sites across North America, as well as 2 affiliated sites. NAMDC works closely with the United Mitochondrial Disease Foundation, as well as with the RDCRN and the Data Management Coordination Center, which is located in Cincinnati. The foundation of NAMDC is its registry. Since its inception in 2011, we've successfully recruited 1,683 patients into the registry across all sites of NAMDC. We performed an initial analysis of the data in the NAMDC registry. This study was led by Emanuela Barca, who was then a NAMDC fellow. Not surprisingly, many of the classic syndromes of mitochondrial disease are well represented in the registry. These include Lee syndrome, knee loss, chronic progressive external ophthalmoplegia, Leber's hereditary optic neuropathy, Kearns-Sayre syndrome, Sandow, Mingi, and others. It is important to note that the 2 most frequent diagnoses were, number 1, multisystemic syndrome, which is defined by disorders which affect 3 or more organs, and number 2, other clinical diagnoses. These 2 leading diagnostic categories point out the need to do further phenotyping to better define these syndromes. Furthermore, the majority of patients with multisystemic syndrome lack a genetic diagnosis. So not only do we have to define these syndromes better clinically, we also need to define them better molecularly. The demographic profile of the patients showed that the age at onset had a biphasic distribution with 2 peaks, one below the age of 2 years, represented by 28% of the subjects, and the other in adulthood, 25% of the patients had adult onset disease. The genetic profile of the subject showed that the majority, 57%, had a genetic diagnosis. Mitochondrial DNA mutations were identified in 39% of the total. Nuclear gene defects were identified in 19%. Most of the patients with mitochondrial DNA mutations had adult onset disease, whereas most of the patients with nuclear gene defects had childhood or infantile onset disease. These findings have been observed in registries in other countries as well. I'd like to call your attention to the gray area of this circle, which shows that 41% of the subjects in the NAMDC registry lack a genetic diagnosis. And many of these subjects who lack a genetic diagnosis have multisystemic syndrome or other clinical diagnosis, again, highlighting the need for further molecular and phenotypic characterization of our patients. This waffle chart shows the frequency of the nuclear gene pathogenic variants in the NAMDC registry. There are 65 different nuclear genes represented in the NAMDC registry. The 2 most frequent were Pol gamma, shown here in red, and PDHA1, shown here in green. However, the majority, 38 out of the 65 nuclear genes, were reported in only single patients. This highlights the heterogeneity and rarity of many of these individual mitochondrial diseases. Here, this figure shows the distribution of mitochondrial classic syndromes and their causative mutations. And as you can see here, Lee syndrome has multiple causative genes associated. The majority of these are nuclear genes, whereas the other classic mitochondrial syndromes, such as MELAS, MERV, CPEO, are typically associated with mutations in mitochondrial DNA. And these mitochondrial DNA mutations often cause more than one phenotype, which is not surprising due to their heteroplasmic distribution and tissue distribution. The phenotypes of these mitochondrial DNA defects can vary widely. The NAMDC registry has been valuable not only for academics, but for industry as well. And here we see a list of the companies that have mined the NAMDC registry data to better understand these complex disorders and aid in the development of much needed therapies. We're happy to partner with companies and share the data in the registry with companies and with academics who are striving to better understand these diseases and, of course, ultimately develop therapies. We have a biorepository, which is housed at the Mayo Clinic in Rochester, Minnesota. We have 377 patient samples. These are our blood samples. The blood is fractionated into plasma as well as buffy coat, and they are available for research purposes. There's a simple application process to NAMDC that will make these samples available. It's been so successful, we've run out of some samples, so we're going to strive to recruit more patients. In addition, NAMDC has a virtual fibroblast with samples from 187 individuals with a variety of mitochondrial diseases. These fibroblast samples are housed in the individual institutions, but are available by application to NAMDC for research purposes. Another project of NAMDC is to define the diagnostic odyssey of patients with mitochondrial disease. The initial analysis was published in 2018, led by Johnson Greer, and it demonstrated that on average, patients were seeing more than 8 clinicians to get to their diagnosis of mitochondrial disease. A follow-up study led by Seamus Thompson and Amel Kara has been completed and is currently in the analysis phase and will hopefully come out soon. NAMDC sponsors pilot projects, including a citrulline dose-finding study for MELAS, led by Fernando Scalia at Baylor, as well as Kipling has been developing a oxygen nanosensor to quantify mitochondrial function in human muscles of patients and controls. Rebecca Ganetsky has a new project to define the role of CA5A dysfunction in complex 5 deficiency. The NAMDC Fellowship has been very successful and led by Richard Haas. Here you can see the 7 fellows who have been selected for this program, beginning with Zuela Gozo-Kipling and Samantha Marin, followed by Austin Larson, Valentina Emanuela, and Emanuel Barca, Isabella Barcelos, and Edward Shadiak will be starting in July. So we welcome him aboard the NAMDC team. I would like to say that all of the graduates of the NAMDC Fellowship remain active in mitochondrial disease, clinically and research. So we're very proud of this program, and we thank Richard for this. NAMDC also sponsors research projects. The project 1 is the NAMDC registry with follow-up longitudinal data and biorepository, which I've already described. And project number 2 is led by Amel Kara, and that is to identify the genetic causes in the molecularly undefined registry patients. As I highlighted before, 41% of the patients in the registry lack a molecular diagnosis, and further genetic studies are needed. Project 3 is a natural follow-up to the genomic studies. Many of the genetic tests will reveal variants of uncertain significance, and to prove pathogenicity, functional studies must be done, and Johan van Hove is developing exactly that, functional studies to assess mitochondrial functions using minimally invasive tissues. Project 4 is a advanced genetic study and a pilot newborn screening study for pyruvate dehydrogenase deficiency, and this is led by Jerry Bedoyan and Suzanne DeBrosse and Doug Kerr in Cleveland. Project 5 is the expanded access deoxynucleotide therapy for TK2 deficiency. As you can see, NAMDC involves a lot of individuals and requires a great teamwork, and we're very pleased to be working closely with UMDF as well as with the RDCRI and NIH sponsors. We had a very robust meeting on Tuesday that lasted more than 5 hours, and we could have gone on even longer to discuss all of our activities. So we are very pleased that we have this opportunity through NIH funding. We're currently ending year 1 of a 5-year cycle. Unfortunately, this will be the end of our 5-year funding from NIH. And so we are looking for ways to sustain NAMDC in the future. I do want to also acknowledge that many people at Columbia, particularly Xiomara Rosales, who's the NAMDC project manager and is really the person instrumental for making NAMDC work. And with that, I thank you for your attention and this discussion. I thank the organizers for allowing me to speak today as the lead investigator of the upcoming Entrada Mingi Natural History Study, ET-NHS 901. Mingi, or mitochondrial neurogastrointestinal encephalomyopathy, is a lethal autosomal recessive disease characterized by mitochondrial abnormalities, including multiple deletions and depletion of mitochondrial DNA, leading to ptosis, ophthalmoplegia, severe gastrointestinal dysmotility, cachexia, peripheral neuropathy, and leukoencephalopathy. In the classic form of the disease, onset is in the late teens with gradual progression over 2 decades, leading to fatality at a mean age of 35. The disease is caused by TYMP mutations that cause severe deficiency of thymidine phosphorylase, or TP. TP resides in the cytoplasm, where it normally degrades the nucleoside thymidine into the base thymine and the sugar 2-deoxyribose 1-phosphate. In patients with Mingi, there are dramatic elevations of thymidine and deoxyuridine in plasma, as shown here, as well as tissues. In the classic form of Mingi, levels of thymidine in plasma average 8.6 micromolar. In contrast, healthy controls and asymptomatic TYMP mutation carriers have no detectable thymidine or deoxyuridine in plasma. In addition to the classic form of Mingi, there's a late onset form with onset in the 40s, with slower progression and prolonged survivals into the 50s and 60s. These patients have milder deficiency of TP and less severe elevation of thymidine with a mean level in plasma of 1.3. Thus, if one could lower the level from the 8.6 micromolar concentration found in the classic form of Mingi to plasma levels of less than 1, one could convert a patient from the classic form to a milder, slower progressive form of the disease with prolonged survival. In fact, This concept has been demonstrated through bone marrow and liver transplantations, both of which have successfully lowered levels of thymidine and deoxyuridine in plasma and led to clinical improvements. Unfortunately, both of these transplant approaches have limitations, including a lack of suitable donors and severe side effects of the immunosuppressive therapies. As an alternative, Entrada is developing a proprietary endosomal escape vehicle technology to deliver thymidine phosphorylase to the cell membranes where they are engulfed by endosomes, which then bud and release the EEV products into the cytoplasm, as demonstrated here in this these immunofluorescent images. You can see the EEV product labeled in red that co-localizes with endosomes. The EEV migrates to the buds where they're subsequently released into the cytoplasm. We've demonstrated efficacy of the ENTR 501 compound, which contains thymidine phosphorylase, using our mouse model of Mingi, which has markedly elevated levels of thymidine in plasma in the micromolar range, similar to the classic Mingi patients. And we can see here that after a single injection of ENTR 501, you can see dramatic decreases of the thymidine level shown in purple to levels that are below the normal levels in mice, wild-type mice, and approaching the levels in normal humans. And this reduction in thymidine can be seen after intravenous or subcutaneous injections and lasts more than 48 hours. In addition, after single injections of ENTR 501 in rats, there was sustained depletion of thymidine for a week. As an initial step in the development of this compound, Entrada is sponsoring a natural history study, which is a multi-center international study, which includes Columbia, Baylor, Colorado, and other sites to be determined. Patients will be followed every 6 months for up to 6 years with a primary objective to evaluate the clinical course of the disease, with secondary objectives to look at biomarkers and additional clinical endpoints and functional outcome scales. With that, I'll end, and thank you for your attention.
Good afternoon. I'm going to give you a very short update on the Mitochondrial Care Network has been doing over the past year. This is a joint effort between the Mitochondrial Medicine Society, MyToe Action, and the United Mitochondrial Disease Foundation. We did launch in 2018, and at that point, we worked on testing each site, assessing its strengths and capabilities. We began some pilot projects. We also began a clinician listserv so that mitochondrial clinicians could share cases and discuss topics amongst themselves. We created a patient tracker to allow for sites to more regularly keep track of their patients and certain patient characteristics. We also began a site needs analysis to see how we might be able to help individual sites moving forward. Our goals for this second year were to form a formal medical board as well as an advisory board, establish a centralized coordinator, receive funding after finalizing a budget, prioritize projects, begin some site assessments, look at new applications, respond to site and patient needs, determine a patient satisfaction measure, and determine patient outcome measures. And we, of course, have not been able to accomplish some of these goals. COVID, of course, hit and put a lot of things into slow motion. So what we were able to accomplish included some administrative tasks. We did form a formal medical board, including co-chairs, Amel Kara and myself. Amy Goldstein remained on the board, and then Carol Green and Irina Ansem joined. We were able to, working with UMDF and MyToe Action, finalize and acquire an annual budget. We were able to post a position for and interview a coordinator, which, depending on if everything goes as planned, we should have completed the hire by the time you're hearing this. And we established legal services, which actually took a whole lot more time than we expected. As far as project updates go, we did release a patient guide for the newly diagnosed, and this is posted on the mitoenetwork.org website. We further evaluated our establishing a telehealth service for mitochondrial disease, and there is a separate update that we provided our group on that. And we continue to move forward with that work. We continue to work with the United Mitochondrial Disease Foundation on a clinician education portal. This is just the front phase of the guide for the newly diagnosed families and patients that's available on our website. And this is the headline for the draft of the paper we are working on, looking at how we assess the needs for a mitochondrial telehealth network and how we move forward with establishing it. There were some projects that were absolutely, unfortunately, stalled for this coming year, including finalizing our research portal, getting a patient needs analysis completed, and looking at our iPad-based functional measure development. And we hope to be able to pick these things up soon. Other unfinished tasks include responding to the site and patient needs, determining a patient satisfaction survey, determining a patient outcome measure or group of measures that we want to track moving forward, opening up for new applications, and then site assessments. Our goals, remember, were to unify the clinicians amongst the group and then work on optimizing and coordinating care, identifying gaps in care, then developing best practices and improving the quality of care to reduce the burden and improve patient outcomes. So what we have looked at is how we might move forward in accomplishing these next steps. And we plan to use this concept of a learning healthcare system that has been established by a group at Cincinnati Children's. It is based on the concept that the current healthcare system is unreliable, error-prone, and costly. It fails to meet patient and family needs and produces unsatisfactory outcomes, especially for rare disease. Moreover, the evidence required to provide care that is safe, effective, patient-centered, timely, efficient, and equitable is often lacking, unfamiliar to providers, or inadequately applied. So the National Academy of Sciences envisioned a learning healthcare system in which the clinical care, science, informatics, incentives, and cultures are aligned for continuous improvement, innovation, and research. New knowledge is captured as a byproduct of care, and evidence is applied reliably and is seamlessly embedded in the delivery process. What do you need to have a learning network? Well, the components include aligning the participants around a common goal, which in this case would be improving care for mitochondrial disease, standardizing processes, policies, and infrastructure to enable collaboration, and creating a commons where information, knowledge, resources, and know-how to achieve that goal are created and shared. And we believe we've at least moved in the right direction for a lot of participants. And we have started work towards step 2 with creation of some governance and policies, as well as starting some network management plans. And then the rest is all on our to-do list. One of the things needed for this work includes a database connected to the electronic medical record, as well as outcome measures that matter most to clinicians and patients, along with a research prioritization committee. And so these are areas that we are going to focus on the most coming up, other than the projects that we've already started. We have not finalized which kind of database we would use yet. Some early work has been done that's looking at the peer platform, which has already been utilized by the United Mitochondrial Disease Foundation and many other rare disease groups, but we still need to do a deeper analysis of whether that's the right database for us or not. We definitely do not want to reinvent the wheel. We already have databases that are collecting data from patients and families for UMDF, as well as from clinicians through NAMDC. And this is meant to be harmonious with that and also patient-friendly and maybe collecting input that we're not already collecting. We will then work on creating a learning commons and exchange so that we can share ongoing work and best practices and provide everybody with regular updates. So moving forward, we should be able to do that, especially now that we have a coordinator hired. So our action plan for now is to finalize the telehealth initiative and to see what we might need moving forward to establish it, update the data from all of our sites, start accepting new applications once our coordinator is fully onboarded, start with our patients' needs analysis, and consider acquiring funding for the iPad functional measure. And then we are going to be forming some committees. The committees are all going to be focused on these high overarching goals, including determining some singular outcome measures that we might be able to better start tracking our patients and how they are doing, determine which research priorities the MCN would like to focus on, look at clinical trials readiness along with the work that Amel Kira has already done with the UMDF, and start seeing how we might best assess each site. And that is our update. I appreciate your sticking with me, and I am available in case anybody has questions. Thank you.
Hello, my name is Dr. Marni Faulk. I am Executive Director of the Mitochondrial Medicine Frontier Program at Children's Hospital of Philadelphia and Associate Professor of Pediatrics at the Perelman School of Medicine. I also have the privilege of being a member of the Scientific and Medical Advisory Board of the United Mitochondrial Disease Foundation. Today, I'm delighted to give you an update on the Mitochondrial Disease Sequence Data Resource, MSeqDR. I have the following disclosures. I am an advisory board member of several foundations and/or company advisory committees. I collaborate, consult, or am PI on multiple different pharma-sponsored initiatives. It is really a pleasure to be able to look back over the last 8 years and think about all that we have achieved as a community in the Mitochondrial Disease Sequence Data Resource Consortium. First conceived in the summer of 2012, we went on to obtain United Mitochondrial Disease Foundation funding and initial funding from the NIH North American Mitochondrial Disease Consortium through the U54 initiative to develop a pilot by which we would create a website that could be a home to genomic and phenotypic data for mitochondrial disease. That website went live in late 2013, and by mid-2014, it was debuted at the United Mitochondrial Disease Foundation Mitochondrial Medicine. Our first publication followed shortly after, and we went on to provide multiple demos at this UMDF meeting in 2015, 2016, and 2017, sharing among our peers and colleagues all the advances that were being made to enable their improved utilization of tools to analyze genetics data, as well as variant information and phenotypic features of mitochondrial diseases. In addition, over this time, many individuals across the world have spent enormous amounts of effort to develop tools that have been centrally accessed from MSeqDR to enable improved understanding of mitochondrial biology and disease. The leaders of the program have also worked to create interfaces and tools to improve the user experience and the ability to have curated data sets on mitochondrial DNA in particular. Of course, the website is not just focused on mitochondrial DNA, but is really enhanced to facilitate nuclear gene analyses as well as mitochondrial DNA analyses. Several years ago, we were awarded an NIH grant from the National Institute of Child Health and Development, a U24 program, to be able to utilize the expertise in our community to curate the genes and variants that cause Lee syndrome. Now, more than 95 causal genes have been identified for Lee syndrome spectrum disorders, and we will present an update to this work shortly. So from our humble beginnings 8 years ago, where we envisioned an enormous amount of data that needed to be organized and collated and made interpretable to our community, we have come a very far way. Today, we are proud to say there are 950 user accounts on MSeqDR with an average page views per month of 6,000. We have built and have made readily available the quick MyToe interpretation platform to be able to analyze genomic data directly by users, including VCF files on individuals and trios to solve the gene that's causing the likely disease in each individual for which data exists. MV tool was launched several years ago in 2018 and now includes reference genomes for 200,000 mitochondrial genomes. A suite of analytical tools, as I mentioned, now exist for analyzing data and variants in both genomes. And most recently, as I'll share with you a few more details on shortly, our lead bioinformatician has created a virtual registry pulling from the literature more than 3,000 cases, of which more than 800 are from Lee syndrome patients, to create the largest in the world case-level registry on mitochondrial disease subjects. We have organized and made available phenotypic data related to Lee syndrome, as well as merging human phenotype ontology terms from major international centers. We have sought and now obtained official approval from the ClinGen NIH program that interacts with ClinVar to create the resources to curate genes and variants, and we have done so as relates to mitochondrial disease. We now cross-link our variants reported in MSeqDR with the broader database in ClinVar. We participate in fair sharing initiatives, and we now can boast fully cross-linked data for 280 mitochondrial diseases for more than 1,600 genes whose products are within the mitochondria or cause a mitochondrial disease, and more than 15,000 pathogenic variants in these diseases. Importantly, we've also created a literature query tool to make it easy for users of MSeqDR to find literature relevant to these diseases, genes, and variants. And really remarkably, over these last 8 years, we have published 14 peer-reviewed manuscripts on work related to this effort, and most recently have published a book that links the knowledge in MSeqDR to a compendium accessible by clinicians and scientists everywhere. The utilization of MSeqDR is truly international, and as you can see, the size of the bubble displays the number of users in each city over a 1-year period. Over the entire 6 years since our website launched, you can see that we've had nearly 63,000 users and 275,000 page views. The number one user is the United States with 41% of all page views, followed by the United Kingdom, India, Germany, and Canada as the top 5. But the number of countries really are global in total. This provides a more detailed look of all of the users in the top 26 countries around the world that comprise the 275,000 views that we've seen, as shown here, over time on this Google Analytic view. What are people coming to MSeqDR to use? Perhaps not surprisingly, it is the phenotypic information that relates to the genetic diseases. So these 63,000 users and 275,000 views, the top 2 pages on which people land are those related to human phenotype ontology and searching human phenotype ontology, the tree features in each of the syndromes that are associated with mitochondrial disease. However, the landing field, understanding the disease features of different genes and variants, and a whole host of tools and genomic support analytic pages are also heavily utilized. We've revised and revamped the landing page to be responsive to users' feedback and needs over time. We have 2 views for the landing page of MSeqDR, which you can see is accessible at mseqdr.org. You can log in as a bioinformatician and scientist or as a clinician, while the same information is available, it is organized differently depending on the user base. And everything is highly interactive. You can hover over all of the white tabs at the top to get to tools, phenotype information, submission of data or variant information, and so on. And you can also come down and either utilize a search for a tool to analyze your own data or a tool to browse the data that exists within MSeqDR. And for those that just want to find all the information they can about a given feature, there's a very forgiving search box on the homepage that shows you in a quick overview all the information related to that in the system. I'm going to be showing you just a few of the more heavily used and recent tools that we have developed in MSeqDR. This is MVTool. MVTool was developed and published by our group in 2018 in Human Mutation, where you can go for more information, but allows you to put in one or multiple mitochondrial DNA variants, again, being very forgiving of the specific nomenclature used. When you put in the variants, it gives you an output as either a text file or a JSON file or a CSV file that you can save that allows you to have each variant and know everything about its allele frequency as it exists in every database, including MitoMap and the different data sets available within MSeqDR, thus helping clinicians and scientists understand the likely pathogenicity of any variant by providing free of charge all the information and all the sources that can be obtained to help clarify what is known. You can similarly go and better understand what has been reported, not just about the allele frequencies, but also about the disease features and the phenotypes of each variant that has been reported. The web interface, as well as the API that has been created to share this mitochondrial DNA annotation data with anybody who would like it, has been successfully used with more than 10,000 runs and 488,000 variants annotated since it has been launched 2 years ago. Similarly, we have a haplogroup calling interface, and this has been used over 18,000 times to annotate 286,000 variants. This information is available to every major laboratory doing diagnostic testing, and thus readily supports their understanding of haplogroups and global information available about any variant they might encounter, thus supporting accurate diagnosis of mitochondrial diseases. Here is another tool that has been a remarkable advantage to have on the MSeqDR website. Thanks again to the work of Dr. Li Shuang Shen, the MSeqDR bioinformatician. You can easily take phenotypic information that you input in either HPO terms or perhaps even a summary paragraph from a doctor's letter with the features in a given patient, click annotation, and select the right terms that match what you would like to describe. You can then upload the raw exome data from an individual or from a family and click annotate, and a file is emailed to you within minutes that will tell you the likely variant that causes the features that you put in for that individual. And so here, for example, you can see that you can upload the files. As an example here, a trio file was used, and you can put in the features that you want and click run. And here is an example of how the report will look in the condensed interpretation view. Upon our trial, it appears that at least 70% of the time, the top variant is the causal disease gene variant in that individual. Thus, it really becomes very worthwhile to utilize by researchers and scientists to try to solve complex or unsolved cases. And all of this data is allowed to be saved in MSeqDR solely for the view of the person who ran the analysis, or it can be saved to their desktop, all in de-identified fashion. Another very exciting new tool that is being developed and is about to be launched is the Mitochondrial Disease Virtual Registry. What this does is creates a registry of more than 3,000 cases from the literature. So all of the literature has been queried in mind as relates to mitochondrial disease, including, as I mentioned earlier, more than 800 patients with Lee syndrome. And a user can come into this tool and click the age, the sex, any of many different phenotypic features, the consanguinity if it's known, ethnicity, age at onset, and so on, and be able to search for literature and specific cases that match that inquiry. This represents the largest registry of mitochondrial disease cases, and again is based upon published peer-reviewed literature. It is particularly useful for Lee syndrome, Leber's optic neuropathy, and general mitochondrial disease as well, and provides both case-level clinical phenotypes as well as genotype data on all of these individuals. It also allows you to break down the information to discern specific clinical features of interest. And what is really remarkable is that it has mapped more than 100 standard terms and more than 110,000 feature records. So this provides a huge resource to our community. Here is an example of what's called the pseudo registry or the virtual registry case information from a subject that was pulled from the literature. And you can see that this individual had symptoms onset at 4 months, passed at 24 months, was in a consanguineous family, was known to have Lee syndrome, was known to have a SIRF1 gene mutation, which is represented here, had these clinical features, and so on. So it really is a remarkable achievement that has taken Dr. Shen many years, and we're excited to see this launch soon. The raw data is remarkably complex, and so the MSeqDR tools had to be built to be able to handle and curate all of this information, a true resource for all of us. In addition to all of the tools for genomic data analysis, variant analysis, and phenotype data, our group has been participating in many consortium-wide activities. In particular, we were awarded in 2017 a U24 grant from NICHD at the NIH to be able to curate the genes and variants that cause Lee syndrome. What we are proud to say is that we were awarded ClinGen mitochondrial disease expert panel status for nuclear and mitochondrial variant curation. The mitochondrial DNA variant curation actually just occurred this week and resulted from several years of working group effort among MSeqDR, ClinGen, ACMG to come up with standardized ways to annotate the mitochondrial DNA genome for any variants. And we're excited to say that work has been prepared for publication, and a manuscript is about to be resubmitted for review. The variants that were piloted in this program were for several genes in which variants are known to cause Lee syndrome and to have actionability that depends on the diagnosis being correct. And these genes are shown here: SLC19A3, PDHA1, POLG, and ETHE1. The group has also been working for several years to curate Lee syndrome spectrum from a gene disease perspective. So while there are now more than 95 genes that are thought to cause Lee syndrome spectrum, more than 60 have now been subjected to expert panel review. We started with a panel of 32 experts from around the world and are now proud to say we have more than 50 experts from around the world who participate in monthly calls and have enjoyed the participation not only of expert curators within our known group, but have also brought on expert curators from industry, including from Illumina and Invitae, who have helped us to review very carefully and present to the group, according to ClinGen infrastructure, the level of evidence that any one gene is or isn't associated with Lee syndrome spectrum. And as you can see here, approximately a third of the genes were found to be definitely causal of Lee syndrome spectrum. 21 had moderate evidence and 19 had limited evidence. And this really teaches us that we all need to continue to publish and share cases in the literature that can be reviewed to help us all understand how likely any one gene or variant is to cause a mitochondrial disease. Another effort among the consortium that has been going on for several years is mapping the human phenotype terms in a defined ontology that comprise the features of mitochondrial disease. For example, Somebody might, in their registry, say dysphagia. Another might say deglutition disorder. Yet another might say poor swallowing, swallowing difficulties, or swallowing difficulty. All of these terms refer to the same issue, which is problem swallowing. And so being able to map how a term is captured in different registries is incredibly important for us to merge data and identify patients who might respond to any clinical trial. And so this work has now been completed for the data that is captured in the NAMDC US registry, the LEMAP registry, the UK, and Australia. So another UK registry as well as Australia. I'm also excited to say that we recently completed a competing renewal application for the U24 grant to expand its scope beyond Lee syndrome to curate the genes that cause all primary mitochondrial diseases, as well as to begin to curate the mitochondrial DNA variants that cause primary mitochondrial disease. And this work is being supported by 50 experts from 15 US cities, 12 countries, and 5 continents. So truly a global initiative. And finally, we have recognized that MSeqDR serves a niche for geneticists and informaticians and sophisticated clinicians to utilize the resources that exist to better diagnose and understand mitochondrial disease. However, to make it even more useful and accessible to the general clinical population, we have now published the first edition of the Mitochondrial Disease Genes Compendium. What this book does is to take a gene-focused perspective of mitochondrial disease, and it provides a readily intelligible, all-in-one reference of known mitochondrial disease genes and their associated conditions, which, as many of us know, can involve any system. The Mitochondrial Disease Genes Compendium has live links to each MSeqDR webpage, and there is where the variant data and tools to better diagnose mitochondrial disease are regularly updated. Within each gene page, the inheritance pattern, the age spectrum affected, the major clinical features, the therapeutics, support groups, and research underway is provided for more than 250 mitochondrial disease genes. Here is an example of one gene as it's presented in the compendium. This gene is a nuclear gene, NDUFS3, and it tells you the information about the gene, the protein function. It tells you the year in which the disease was first discovered, and it gives you fully referenced information where you can go to gain more information. It tells you which genome it's in, the inheritance pattern of the disease, and remarkably, we have more than a dozen genes that can cause either autosomal recessive or autosomal dominant disease. It tells you the number of pathogenic variants that are known or that are uncertain in the ClinVar general database. It tells if there's any common mutations that have been identified, any disease names, the age at onset in which symptoms have been reported in the literature, the specific manifestations that have been seen by organ system. So here, central nervous system, muscle, ophthalmologic, pulmonology, GI, skeletal, and metabolic features. Every gene has a unique clinical Pearl section that is particularly helpful to understand the scope of the disease. Any signatures that might be present in blood, muscle, or other tissue, natural history studies, specific therapies, clinical trials, a list or a link to what model animals might have already been reported for that gene, and which advocacy groups exist to support individuals diagnosed with this gene disorder. It also provides a link to the MSeqDR gene card, as well as a list of the authors and the editor for each gene. And here on the right is what you'll see when you click on this link. You can easily see all of the information that can be found in MSeqDR and can be clicked on to take you wherever across the internet more information lies about the protein, the publications, the variants, the disease features, and so on, all the variants in that gene, and so on. So we are hopeful that this will be a very easy to use system to provide general information that's accessible in the clinic or in the laboratory, and then to be able to click from the ebook version directly into MSeqDR to get to the page with more of the information that is timely and up to date. So how can you join? We continue to have for the last 8 years, and will continue to do so, monthly MSeqDR Consortium meetings. It is the last Monday of each month at noon Eastern Time, and that is where we provide updates on new platforms, new tools, new activities, and we request feedback from consortium members. We also have regular U24 project meetings to do gene curation and variant curation, and we're always happy to have new members join us. And to do so, please contact Elizabeth McCormick at the email shown here. And finally, MSeqDR exists to support our users, whoever they are. It's a very diverse user set, clinicians that are genetics experts, clinicians that are genetics novices, general scientists, biologists, and so on. And there are tutorials available on the homepage at mseqdr.org. Finally, we strongly encourage you to contact Lishuang Shen, who has been the creator and lead bioinformatician of MSeqDR since its inception, with any questions, comments, or feedback. I'd like to acknowledge the development team who has put in countless volunteer hours over this last 8 years to make MSeqDR a truly useful resource and to support the community, recognizing that we've had more than 100 other individuals make major contributions to the content and the function of MSeqDR. A particular call out to Dr. Zhao Gai, my co-leader of MSeqDR, who's at Children's Hospital of Los Angeles and has been developing a suite of tools that are still coming down the pike to support our better understanding of mitochondrial genetics and disease. Lishuang Shen, whom I mentioned, is our outstanding bioinformatician, really responsive to our community needs and working tirelessly to provide more user-directed, improved content. Dr. Zuela Zokapli has been a leader in the human phenotype ontology effort. Elizabeth McCormick is our lead MSeqDR study coordinator, works very closely to integrate our activities with the United Mitochondrial Disease Foundation, and is our lead coordinator for the U24 program. Colleen Murescu as well is a genetic counselor who helps us enormously with the MSeqDR genomic data handling process. And of course, Dr. Phil Yeske, the Science and Alliance Officer from the United Mitochondrial Disease Foundation, has partnered with us and attends our regular working group meetings to continue to make MSeqDR useful to support the activities of the global mitochondrial disease community. As I mentioned throughout, the funding has been consistent from the United Mitochondrial Disease Foundation to support our work for these last 8 years, as well as intermittent funding from the National Institutes of Health, including NHGRI, NICHD, and NINDS. And so I'm grateful for how far we've come, and hopefully we will continue to be able to support mitochondrial biology and disease research for many years ahead. Thank you.
Hello, everyone. This is Philip Yeske, UMDF, back with you to provide the final infrastructure update of the day. I'll be speaking about the Mitochondrial Disease Community Registry. If you recall, this was a UMDF initiative that we began in 2014. And our goal was to create a novel participant-centric registry. And our partner, software partner from the outset of the project and continues today, is Genetic Alliance. They were the developers of the platform for engaging everyone responsibly, or PEER. So since 2014, we've recruited 1,500 unique individuals to the registry, and they've created approximately 2,400 profiles. And through those profiles, and by presenting a survey to them, we've collected a limited data set that had some health information, some diagnostic information, again, from the patient perspective, but also a number of free text questions where we had the opportunity to ask for opinions and perspective on important topics like participating in clinical research. What's the most important thing that you would want a researcher to know, or a regulator to know, or a drug developer to know about living with mitochondrial disease? And many of the findings through that time echo what we heard at our patient-focused drug development meeting that we hosted in March of last year. But it also amplified upon it. It was really an opportunity to gather a lot of those perspectives. So to put a bow on the first 4 or 5 years of the registry, I started working with a volunteer data analyst, Sophia Zilber, to co-author a paper where we summarized these findings. And some of the interesting things that came out of it is that while many of the patients who provided answers are hopeful about the future and our ability to develop treatments and cures to treat mitochondrial disease, there were also concerns expressed about, or the challenges associated with interacting with clinicians or feeling alone and disconnected, right, from others. The paper is shown here on the screen. You can find it on the website for the journal, Mitochondrial and Metabolic Medicine, or you can come to the UMDF website, and at umdf.org/registry, you'll find a link to download a free copy of the paper, and it's free to share. I really encourage everyone listening today to do that. It really provides nice insights into what is of greatest concern to our patient community, which I think is really important as we try to advance our mission with a specific emphasis on developing treatments and cures for mitochondrial disease. So the rest of my talk, I'd like to focus on what we're trying to pivot to now with MDCR. And it's a really exciting time. In 2019, Genetic Alliance entered into a strategic partnership with a company called Luna DNA, shown here. And they began the effort of completely rewriting the code for PEER. So this is a brand new registry database, brand new code. And we're going to narrow our focus on MDCR now to a singular goal. And that's to facilitate the development of treatments and cures for mitochondrial disease. We asked, we listened, and this is what the majority of people say they really want to come out of whatever data sharing they're doing. It's not to say there won't be other opportunities to share other types of data or opinions, and I'll talk about some of that later. But the primary goal of MDCR with the launch of this new version of PEER this summer will be facilitating the development of treatments and cures for mitochondrial disease. So how do we do that? That's a talk in and of itself. But if I had to reduce it to a couple of bullet points, first and foremost, it's about identifying and characterizing mitochondrial disease patients, again, from the patient perspective, sufficiently to be able to enroll them into clinical trials or find clinical research opportunities that are a good fit for them. With those patients in the database, we'll then be able to build robust natural history data. And again, natural history tells us the progression of the disease over time. So using patient-derived data, we can build out complementary natural history to any clinician-derived natural history. I might take a moment here just to comment further on that. A reminder that earlier today we heard from Dr. Michio Hirano discussing NAMDC, which is the clinical research network and clinical registry associated with it. So that's clinician-derived data. MDCR provides us patient-derived data. They're complementary to each other, not competitive. So with the Luna PEER platform, which is how I'll refer to it here going forward, you see the process shown. Of course, it begins with identifying, engaging, enrolling the patients in. UMDF plays a large role in that. And then Luna really is what, with their platform, is driving the collection of the data, the management of the data, and facilitating research. Ultimately, though, it's the responsibility of Genetic Alliance in partnership with UMDF to continue to engage the community and drive more people in. So this is all a big cycle of trying to build more and more data. What I really want to emphasize here, though, is that UMDF takes very seriously its stewardship role in this project, and that extends from beginning to end. It's our responsibility to ensure we're appropriately engaging the patients, that we're collecting data in an appropriate way, that we're presenting research studies that are appropriate to them, and that we're creating value and valuable content and continuing to engage those registrants over time. So let me talk a little bit about how the new platform will look relative to the old one. So immediately, you'll see that one of the common themes from the current platform to the new platform is emphasis on member control. That is to say, as a registrant, you are in full control of your data. Your data is never sold. Any research that's conducted is used in de-identified data. You can delete your data at any time. You can revoke consent to participate in research at any time. And ultimately, if you no longer want to be a part of the registry, you can delete your account at any time either. So this is all focused around privacy and ensuring that those who provide the data are in control of their data. From a data perspective itself, the Luna peer platform will have some very nice capabilities relative to the existing platform we've been on for the past 5+ years. You'll be able to attach DNA files, initially from organizations like 23andMe or Ancestry.com. So simple DNA files, but this really helps to understand the genetic underpinnings of mitochondrial disease for each individual that registers. We'll also be able to request and attach your electronic health records. So there's a whole process built into the platform to facilitate outreach to the institutions where you get your health care, request your health records, which you have every right to do, and then bring them onto the platform where they can then be used for research purposes. Of course, there will still be surveys. Luna has a standard set that cut across all disease communities, but we'll also be able to develop customized surveys for the mitochondrial disease community. And finally, studies. Think here like a natural history study for a specific form of mitochondrial disease. So many types of data from the get-go when this launches this summer of 2020. But in the near future, there'll be even more data types. And this really helps to broaden and deepen the registry in terms of content. Additional DNA data types, many of you will have had panels or whole exomes conducted, or even a whole genome sequencing done. You'll be able to attach those data to the registry, to your account inside the registry, and then consent to the use of that for facilitating research. Wearable data. So an example might be like a Fitbit, so that we understand how movement and daily activity affects mitochondrial disease. Medical devices, medical imaging, many other types of data are also under consideration. And I think the message here, this is a platform that's really poised to grow over time, broaden the data, deepen the data. Lastly, let me talk briefly about what we do when we have the data in. Of course, very limited utility if we collect data and don't use it for any purpose. So a very nice component of the Luna peer platform is what they're calling the sandbox. And this is where there will exist a robust set of tools for analyzing the data and conducting research inside the platform. That is to say, the tools are available on the platform, the data never leaves. So for the researchers and clinicians that are listening today, please know that our patient-populated registry is really moving into a new phase where we'll be able to drive studies, collect data, and conduct research all on the same platform. So there'll be much more to tell about that. I'll end there and just say that if you are interested in participating in MDCR and you're not currently a member, we do have the registry down right now because we're transitioning to this new platform. There is a page, which I've shown here on the screen, umdf.org/registry, where there's a link available to be contacted whenever the registry is open again and accepting new registrants. If you're a current participant in MDCR, first of all, thank you. We appreciate everything you've shared to date. We'll be in touch. There's no steps for you to take at this time, but we'll be in touch soon to talk about the steps necessary to transition your account from the old peer platform now to the Luna peer platform. And I think you'll be really excited when you get a look at the new platform. Please stay tuned for more details. Our estimation is sometime this summer we'll be in the full launch of the new registry. Thanks for listening today and have a good day.